Hit the music!
Drip, ça fait là, ah. Drip, ça fait là, ah. Drip, ça fait là, ah.
Oh, I do love a bit of McFly. Hello, it's the 9th video in the Evolutionary Ecology series. Now, the bit of the series that we're in at the moment is all about the limits to adaptation, and a lot of it is based around a very theoretical framework, right? And that means that all these different videos that have been coming out lately kind of merge into one, really, because basically what we're going to be doing today is picking up from where we left off. But this time, we're going to focus on limits of adaptations on whole wild populations. And this is all about stating that the local selective environment on organisms is particularly important. But then that creates a sort of a paradox, if you like, because if you go out into nature anywhere, you see that there's a lot of variability in nature. Within a single species, organisms vary, okay? So if evolution is so strong, surely it's better to converge on one single form, okay, through a process of purification, right? So why do we see all this variation in nature? So perhaps at any one time there are loads of phenotypes that we see in the population which aren't actually particularly fit, okay? And we can get some indication on whether that might be the case by looking at rates of evolutionary time and comparing them to how they are now to how they were in the past, okay? So if we go back in time, the evolutionary rate seems to be a lot slower than it is now. So this may suggest, as we've been talking about in the last video, that for long-term evolution, a much smaller amount of variation is suitable to create big changes, okay? I mean, if we think about things like artificial selection in our domestication of crops and livestock, then we've removed all these ecological trade-offs, and as a result of that, evolution is a lot quicker. And this, coupled with these extremely strong selection pressures in artificial selection scenarios, means that traits get selected for a lot quicker. There's less constraints on their evolution compared to in wild populations. Now, when measuring the rate of evolutionary change, the unit we use to measure it, unsurprisingly, is called the Darwin. Okay? So the rate of evolution currently on a particular trait, whatever it may be, is greater in the number of Darwins now than it was before. Okay? Now, that could be because most mutations are deleterious. So only a small proportion of the mutations that are occurring at the moment will have positive effects. Another thing is when you look at the fossil record, all the fossils that we've got from a particular species or whatever are usually located in one geographical location. Okay, so it's only given a small subsample of the entire range of a particular species where a lot more evolution could be going on. Okay? Another thing, and probably the most important point to mention here, is the unpredictability of the environment. Okay? The environment is constantly changing all the time. From one day to the next the environment is different, so let alone over millions of years of evolution. Okay? So that creates a paradox of stasis in the fossil record, because all the fossils that you get are from presumably more or less the same time period. So all you're getting by looking at the fossil record and the phenotypes you see are the averages. Okay? You don't see as much variation. So this may suggest, as we suggested at the start, and highlighted by Ginnerich in 1983, that some phenotypes are relatively unfit and are therefore doomed to extinction at some point, which is a very dramatic way of putting it, isn't it? Now, more of these evolutionary studies have been done on Drosophila melanogaster, this time done by Barton and Keatley, I don't know what it is, in 2002. And what they did is that they artificially selected for a particular trait. whether that's bristle number or something else, right? And what you see is that when you have this artificial selection on these fruit flies, then there are phenomenal rates of adaptation. Okay, so traits can be selected for really, really quickly. Now this is sort of giving the impression then that there's a lot of variation that evolution can act upon. Okay, and if you remove this selection pressure, then these flies can return to their normal phenotype. Okay, but they won't completely return to their normal phenotype because some alleles would have been lost along the way. Okay, so you could almost describe that these Drosophila fruit flies have gone through an artificial genetic bottleneck, if you like, and the result of that, they've lost some genetic diversity. So when looking at all these examples in the wild though, we must consider all the environmental effects within and between generations. Okay? And of course there are ecological trade-offs, but only a proportion of these are labile. And this becomes particularly important when the environment is harsher, when there's a greater selection pressure on organisms. Okay, that becomes particularly noticeable. Now, we've been talking about pleiotropy more times than I care to think about in these past 3 videos. Another thing we need to consider is physical linkage. So this is when mutations arise on a chromosome which could be beneficial, but those mutations are physically linked to mutations which are actually deleterious. So that weakens the selection pressure on these beneficial mutations. Okay, so this all leads to the idea that alleles do not work independently because they're physically linked to other genes on the chromosomes which have different effects on fitness. Okay, so it's a difficult world being a gene that wants to be selected for. So therefore genetic variation must be substantial in the long term, but this doesn't necessarily mean that there's high variation in fitness. And a good example of this idea was undertaken by Ettison and Shaw in 2001, where they showed trait correlations can constrain adaptation to factors such as climate change. Okay, and we'll also, by the way, have a whole video on how climate change has influenced evolution of species later in the series. Okay, so this was done in a leguminous plant, so in the bean family. Chemicrista fasciculata was transplanted across a latitudinal gradient in the USA. And they watched to see how they got on. And what they found was that these trait correlations that we see actually can assist adaptation in some areas, but hinder it in others. Okay? So the trait correlations weren't consistent across populations. Remember again, the variance in fitness is much lower than the variance in the trait due to all these pleiotropic effects. But the key message here is that these weren't consistent across the populations. There's a huge load of constraints to the adaptations of these leguminous plants, right? So there's a lot of variation in a trait, but that variation is not necessarily in the direction natural selection wants to go in because of all these constraints such as physical linkage and pleiotropy and whatnot. Okay, so that creates a huge problem and slows down selection. And you can do this by calculating the selection gradient. Okay, because if you look at the selection gradient, you see that it's not as good as it could be. Instead we calculate this parameter here called the G max, which is the line of least resistance, okay, which eventually reaches the optimum, but it's not the quickest route because of all these constraints. This brings us on very nicely to the idea of the visibility of genes to selection, where there's different levels of these trait correlations in good and bad environments, okay? Because of course, traits also interact with the environment. Okay, so although it's likely that selection is going to be stronger in natural environments, the environment can slow down the response to it. And some nice work has been done showing this by Crook and Pemberton in these two different animals, red deer and soay sheep. Now if you were a male stag on the Isle of Rum, which is where Crook and his colleagues studied these deer, then having big antler size is a good idea. Not only is it good for fighting with, but it's also good to impress females. Okay? But what is interesting though is that we don't actually see a selection for increased antler size over time. And the reason behind that is that antler size is actually condition dependent. Okay? It actually depends on the condition of the male. So that means then that it doesn't matter what good genes you have, they need to be in the right individual, if you like. Okay, so that red deer stag needs to be in the right environment, need to have been brought up in the right way, needs to have been fed a lot, so it can build muscle and whatever it needs to fight. Okay, for that trait of antler size to actually be any use, to be properly expressed. Okay? There's also a thing that increased antler size can also be damaging to females as well, but we won't go into that. Now for soay sheep in the Hebrides where they have really harsh winters, it's important to be big. Okay? So you have enough fat reserves to last you through the winter. Okay? So you'd think that it would be more adaptive for there to be greater variation when conditions are harsher. Okay, because that means your offspring have a greater chance of survival, or some of them surviving. Okay, but actually calf size variation seems to be greater in good years, and this is because of the interaction with these traits and the environment. Therefore, the environment is masking a lot of genetic variation that we see. These genes need to be in the right place at the right time, if you like, to be fully made use of. Okay. Another thing to take into account is that cumulative chance of non-genetic death increases with age, and that dilutes the selection pressure on these traits. This coupled with the idea of phenotypic plasticity that we talked about in the first video, where the genotype and the phenotype don't always align with each other, they differ, then this all leads to the idea that sometimes good genes aren't always useful. Okay? They've got to be present in the right environment and in an organism of the right condition, as we saw in the red deer. Okay? But this cumulative chance of non-genetic death is quite interesting because it brings us to the idea of the evolution of aging. Okay? I'm gonna die, you're gonna die, everyone on this planet is gonna die at some point. Why is that? Okay? And we can explain all of that by talking about this conflict that's going on within our own genomes. So this is all about trade-offs within the genome at different life stages. Now we've already stated that the cumulative chance of random death increases with age, right? Sorry, that's a bit of a depressing statement, but yeah, there you go. And because of that, there's a reduction, there's a dilution in the selection of traits as you get older, okay? So there's less of a selection pressure on traits which are beneficial when you're older compared to when you're younger. Now this can get particularly extreme where you have one trait, one gene, which is beneficial to you when you're younger, but actually is deleterious to you when you're older. And because it's beneficial to you when you're younger, that means it's going to be selected for, even if it can increase your risk of death when you're older. So this idea of pleiotropy can be antagonistic. We have antagonistic pleiotropy going on here. And this is because selection isn't strong enough to break up these negative genetic correlations, okay? Because there isn't a strong enough selection pressure, okay? And this is likely to be the case in the evolution of all life histories. Now, We can experimentally test for this. Obviously we can't do that in humans because that would be quite unethical, but we can do it in Drosophila. That's absolutely fine, isn't it? So we can artificially select Drosophila for a greater longevity. Okay? And what we find was over, I think, 5 to 6 generations, you get a 30% mean increase in lifespan if you select for greater longevity. So this is artificial selection for longevity. However, if you increase longevity, it was found that there's reduced fecundity and egg size of these Drosophila. Okay, so this is the trade-off. There are negative consequences to living longer. So the Drosophila, I don't know how long a Drosophila lives for currently, I'm sorry about that, I know you were dying to know. Whatever it is, its life history is the output of this trade-off between longevity and fecundity. And similar studies have been done in humans. It's been suggested that an increased levels of testosterone in males increases the chance of prostate cancer later on, and increased levels of estrogen in females increases the chance of osteoporosis. So obviously these hormones, testosterone and estrogen, are very beneficial when it comes to reproductive success, but the more they're produced, the more negative consequences there will be to come, I'm afraid. So that means there's selection acting on traits within an individual's life. Okay, so that's something we haven't really come across before. So basically, just to conclude then, there's this engineered redundancy where selection delays negative consequences rather than banishing them completely, because the selection pressure as you get older isn't strong enough to do that. Right, well that's a pretty depressing way to end, isn't it? We'll come back next time where we'll finish our speculation on the limits to adaptation. So I'll see you then.